Okay, so today I have the pleasure to tell you some of the research that my lab is doing at the EMBL in Heidelberg, where we focus on developing new technologies for imaging in the life sciences in general. But here in particular, as the topic of the talk suggests, about developing techniques and tools that allow us to observe neurobiology in the living organism. Okay, so as I said, one of the major aims of our lab is to advance intravital neuroimaging technologies in the living mouse. And this is, from a microscopist point of view, a daunting task, because the scales that we have to bridge here in the spatial, but also in the temporal domain, span many, many orders of magnitude. And so in the field over the years, multi-photon microscopy has become one of the gold standard tools. And many in the field, and also us, we are trying to improve its performance by increasing the field of view, maintaining or improving its resolution, but most importantly in our lab, trying to increase the imaging depth, which is important if we want to capture neural function and structure in the physiological context of the entire tissue, here the brain. And so today I will tell you about some of our efforts in which we are trying to increase the imaging depth in multi-photon microscopy, specifically in the mouse brain, while maintaining diffraction-limited resolution. And so if you want to image deep, we have to realize that we have to overcome the problem of light attenuation, which is both due to absorption and scattering inside the brain. And what I've plotted here is both absorption and scattering, but the important curve is the green one, which is the attenuation length inside a mouse brain. And the higher this length is, the deeper we can basically penetrate. So now the problem is that normally two-photon microscopy is being done at wavelengths of 8 or 900 nanometers. But as you can see from this plot, the maximum is being reached at actually 13 or 1700 nanometers, which, if we want to use fluorescent proteins in the visible, we have to go to three-photon excitation. But first of all, this shift in wavelength already allows us to overcome some of the problems, especially shifting the excitation from 800 to 1300 nanometers already almost doubles the penetration depth in brain imaging. But it's not just about the shift in wavelength. This was still being done with two-photon excitation. The three-photon excitation also enables us to improve the signal-to-background ratio, as you can here see nicely from this work that was being done in the Schul lab. And you can see really this marked improvement in signal-to-noise ratio, which is due to the fact that the out-of-focus fluorescence that happens far away from the actual excitation spot is markedly reduced in three-photon microscopy because of the higher nonlinear excitation. However, it's not just about depth, it's also about maintaining good optical resolution, which is a big challenge in the field, since optical aberrations, both from the microscope itself, but also from the different interfaces, such as the immersion liquid here, the cover glass that we use as our brain windows, and the tissue, all induce various sorts of optical aberrations. Now, luckily, active wavefront correction methods such as adaptive optics exist that allow us to pre-compensate these aberrations, and therefore lead to the fact where we can focus to the smallest possible point inside our sample, in our case the brain. And realizing and maintaining very good focusing is important for two reasons, especially if we want to visualize and resolve fine structures in the brain, such as here the dendrite with its various synapses, which are on the order normally of about a micron or even less. And so here an improved point spread function due to adaptive optics helps us twofold. A, it increases, of course, the resolution because the point spread function gets smaller, but also it leads to a more efficient excitation of small structures that are smaller than the point spread function. So our goal was in this project to combine the before mentioned techniques, three-photon microscopy and adaptive optics, to chip away at this infamous pyramid of frustration that probably most of you have dealt with in microscopy, where it's difficult to push one of these parameters without affecting the other. But by using new approaches, we can avoid some of these trade-offs And so we wanted to use three-photon microscopy to increase the depth. We wanted to use adaptive optics to restore the diffraction-limited resolution that is often lost in deep brain imaging, and all of this while ensuring that the mice remain in a physiological condition. So first about our three-photon microscope. This is actually a project that started more than 6 years ago in my lab, and Lina, my first PhD student here, set out to design the system that can be summarized by this very simple schematic here on the left. But I also want to highlight that there's many different parameters that we need to tweak and optimize in three-photon imaging. Most importantly, the lasers need to be chosen such that the excitation probability is as high as possible. And for this low repetition rate, lasers with very short 50 femtosecond pulse duration are optimal. But I just didn't want to show just one of these typical schematic illustrations. I also wanted to show you the setup as we have it at the EMBL, which is of course a much more intricate system. And I'll talk about the various elements here that are important in a minute. But before doing so, I really wanted to showcase the improved imaging depth that can be afforded by three-photon microscopy. And here I show you an image stack in the mouse visual cortex that starts at the surface here. And normally with two-photon microscopes, we can reasonably image down to around 600 micrometers in the brain. But now with three-photon microscopy, we can more than double this imaging depth, going not through the entire cortex, but also through this highly scattering corpus callosum layer and then into the hippocampus. And as you can see in some of the zoom in or individual image planes, the signal and contrast is really great here in layer 6 at around 700 microns. But also further down here we see the corpus callosum fibers and also into the hippocampus. And all of these images hopefully show that the three-photon modality really yields a high contrast at large tissue depths, so before this onset of out-of-focus fluorescence. However, we normally have to take great care in intravital imaging to not blast the brain with too much light excitation, and that's usually a big concern in the field. And so we've expended some time to optimize all our imaging conditions to make sure that the brain remains in a healthy and viable state. And so for this, what we normally do is that we first characterize how the brain attenuates our fluorescent signal with depth. So we fit these curves to the various brain regions. From these curves, we can then calculate what the exact pulse energies are at the focus at the various depths. And what you can take away from this graph here is that our pulse energies at the focus all stay below commonly accepted thresholds that have been previously established in the field. Now the next challenge that we are facing when we are trying to resolve subcellular structures such as dendrites and synapses in the brain is actually tissue motion, mostly due to heart and vessel pulsation, as you can see here in this movie. And this is a problem because normally what we are doing is we are trying to image several frames and then to average them. But if the tissue moves, then of course we cannot really do this, and this problem really becomes more and more pronounced with increasing image depth. So we decided to tackle this problem by actually recording the ECG signal that from the mouse, and then to actually program an FPGA to synchronize the scanning of our microscope to the ECG signal, so that the images would only be acquired during the resting states of the heart, so when there was no active tissue motion due to heart pulsation. This actually works fairly well. As you can see in these two movies here, you can see this intraframe artifacts here, these pronounced wiggles when we switch off the ECG gating. But with the ECG gating, the images are much more stable, as you can also see in this more quantitative analysis where we sort of plot the correlation of one image to the subsequent images in time. All right, so I told you how we can achieve large image depths with three-photon. I told you how we can keep our mouse brains happy and stable. So now I wanted to devote most of the time to adaptive optics, which is by far the most challenging aspect of this work. And so the main challenge in correcting aberrations is to actually measure them in the first place. And so for this, there exist two approaches in the field. One of them is called direct wavefront sensing, which uses a camera and a so-called Shark-Hartmann sensor to directly detect the aberrated wavefront of the fluorescent light. And this has the advantage that it's of course extremely fast because it can be just imaged on a simple camera. But the disadvantage is that it requires ballistic light to actually reach the camera, and this can quickly get overwhelmed by tissue scattering, as you can see by this work here, where dependent on the wavelength being used as a guide star, this limit is being reached between 500 or 700 microns. So certainly much earlier than where we would like to do adaptive optics. The opposite approach is a so-called indirect method, where in contrast we don't try to actually image or sense the aberrations, but where we apply the corrections directly to the excitation light, and then only cycle through different modes until we found the optimal correction pattern. This has the advantage, of course, that it's independent of the wavelength, because normally in direct wavefront sensing methods, you sense the aberrations at the visible fluorescence light, but then you need to apply the correction in the infrared excitation light. But also more importantly, we found that the indirect methods are more robust to low signal to noise ratio, which we typically find in deep imaging, and that therefore we are also less depth limited. So how does this work? Normally we have a deformable mirror that can change the optical modes, and then we cycle through different modes. Here in our case we chose a so-called Zernike modal approach, where we decompose the light patterns into distinct Zernike modes and then optimize for each of them individually. And this has the advantage that at least ideally these Zernike modes are orthogonal to each other, and therefore they can be sort of independently optimized. Now the advantage here is also, since the modal adaptive optics allows you to independently optimize them, is that the fluorescence only needs to be sort of stable over around 9 measurements, which we take for every single mode to then fit and find the optimum. And since we are doing this at 25 Hertz, it only takes us a fraction of a second to actually find the correct aberration correction. Okay, so how well does this work, especially in vivo? Here you can see an image being taken without adaptive optics at around 800 microns. Now, if we switch on the adaptive optics, of course, we markedly improve the signal, but we also improve the resolution, as you can see here when we zoom into one of these dendrites, where at the same imaging conditions without adaptive optics we can't, but with adaptive optics we can resolve nicely the dendrite and the synapses. And we can also perform line plots through our dendrites to measure the axial resolution. As you can see in these plots, this allows us to halve or double the resolution in this case. This is a twofold improvement. We can also image through the corpus callosum and into the hippocampus and observe large signal improvements due to adaptive optics, both on the soma, where we typically observe a threefold increase due to adaptive optics, but especially in dendrites or smaller structures, where we can get much larger signal improvements. And this is not so surprising if you remember one of the introduction slides that I showed, where I said that especially for structures that are smaller than the point spread function, the improvements are expected to be more pronounced. But we were actually able to go much deeper than the cortex, and here I'm showing you some data from the hippocampus. These are axial projections, where we are optimizing the adaptive optics here on the single cell soma in the CA1 layer of the hippocampus. And still with this correction, we are able to still get a large improvement of the image, of the signal and image quality at much larger depths, here about 1400 micrometers. So these are all nice images, but we also wanted to actually show and analyze that the improvements in resolutions are really quantitative across the whole image. And so for this, we analyzed the frequency components in this image, a so-called spectral power analysis. And here you can also see that both in the cortex and the hippocampus, whenever we switch on the adaptive optics, we get a marked increase in the spectral power and in the spectral frequencies, which points to the fact that we're really improving resolutions throughout the image. Now we can also do again, this axial resolution quantifications throughout depth. And here what you can see is that when we switch on the adaptive optics, we can almost restore the diffraction-limited resolution at almost all imaging depths. Only at around 1300 microns, we are not quite able to get to the diffraction limit. But now, since we are capable of performing adaptive optics over really the entire cortical column and even into the hippocampus, this allowed us to study and characterize the aberrations and how they actually spatially vary in the brain. And so for this, Lina assembled really a comprehensive data set. Here you can see it in the layer 5-6, but we did this across the cortex and the hippocampus, where we performed adaptive optics correction at one site, and then we switched the adaptive optics on and off, and we quantified how big the signal improvement would be. And then from this we can analyze how the adaptive optics improvement and therefore correction works as a function of distance from the correction site. And what you can see here, also dependent on the Zernike modes, or the number of Zernike modes that we chose to analyze, is that the isoplanetic patch, so the field of view of which a correction is valid, is on the order of around 150 microns in the lateral dimension, but in the axial dimension it can actually be much, much larger. And that was actually very surprising for us, because it has not been reported before in the literature. So here you can see just a more summarized plot here in the cortex and the hippocampus. So the isoplanetic patch here falls off and is around 150 or 200 microns from the correction site in the cortex, but in the hippocampus it becomes actually much, much larger. And I think this points towards the fact that the aberrations are being mostly introduced higher up in the imaging path, and that the sort of the excitation light cone is less changing and affected as we then actually translate deeper. And this is an important finding, because it actually shows us that we do not need to correct for the aberrations in a very tightly sampled fashion. We can actually use just a few cell soma that are on the order of 200 or even further apart from each other, perform the adaptive optics measurements there, and then use these corrections for a large field of view. We also learned in our study that the aberrations actually increase in depth. This is maybe not that surprising, but what we found is that that adaptive optics in this deep brain region, especially hippocampus, require a stroke, so a maximum amplitude of the deformable mirror that is larger than many of the first models could handle that were out there. All right, so next we also wanted to show that we cannot only image structure, but also function with our three-photon adaptive optics microscope. And so for this, we teamed up with the lab of Amit Agarwal, who studies astrocyte biology inside the brain. And here it's well known there exist two cell types, the so-called protoplasmic and fibrous astrocytes, and especially these later astrocytes, the fibrous ones, reside in the highly scattering corpus callosum layer, and they are typically too deep to be imaged with two-photon microscopy. So here the goal was really to use our microscope and to be able to image these really deeply lying astrocytes. As you can see here from a protoplasmic astrocyte at 800 micrometers, the quality again is good enough that we can image also the functional dynamics and analyze the so-called microdomain calcium activities in this case. But more importantly, we were also able to perform calcium imaging of the fibrous astrocytes, as you can see here on the right, and also quantify their calcium dynamics over time. And since astrocytes are very light sensitive, this is really where adaptive optics come into play. So this is a recording where the adaptive optics at first is off, but then you see we switch it on and we get marked improvement in signal. And this improvement in signal, of course, also allows us to identify and detect a lot more microdomains. Here in this case, almost a 6-fold improvement in the number of the detected microdomains. And again, because these glial cells are very light sensitive, adaptive optics here helps to keep the overall excitation powers low and still get decent signals in this deep brain regions. Well, to further then highlight the potential of our three-photon microscope, we also teamed up with a second lab at the University of Heidelberg, with the lab of Varun Venkataramani, who together with others in Heidelberg is studying glioblastoma or brain tumor progression. And here it's well known in the field that both in humans, but also in the mouse, the corpus callosum, so the white matter, is an important invasion route for this disease. And in fact, many clinicians actually say that glioblastoma are a white matter disease. But again, the problem here was that so far, many of the people in the field were using two-photon microscopy to study only the cortical regions of these brain tumors. And so it was not possible to actually study the cell migration, infiltration mechanisms at the more pathophysiologically important corpus callosum or white matter brain regions. And so here we wanted to set out and use our microscope to actually look at the different cell migration patterns that this cell display. But first of all, we realized that if we wanted to do cell migration imaging deep inside the brain, we need a very stable, very robust microscope. And we also realized that very often the labeling of these glioblastoma cells is much lower, so the fluorescence index is much smaller than with the previous lines that we have been using. And to tackle that challenge, we turned to image denoising, which of course is a technique that allows you to basically make the best out of your photon budget and to improve the image quality and therefore enhance the ability to analyze the images, or to turn it around, actually use lower power to observe the same image quality. However, we also realized that some of the available tools, such as noise to void, that are, of course, already exist in the field, do not really work very well on our three-photon data. And this is mostly due to the fact that the noise sources in three-photon microscopy are slightly different from two-photon or one-photon data. And so for this, Emmanuel, a master student in my lab, came up with a special three-dimensional version of noise to void, and he also programmed a so-called custom structural noise removal that sort of gets rid of a lot of the three-photon noise data. And so this works now fairly well, and as you can see here on the left in these movies, but also on the right in the quantification, the image denoising here really affords us to increase the signal to noise by about 20 dB across the whole imaging depth. Of course, it's mostly very important at the deeper layers where we wanted to actually image the glioblastoma cells. And so this image denoising also brings me to a very important technical point that I really want to highlight here. And this is the fact that although denoising improves the apparent SNR, it is not a substitute for missing resolution and therefore for adaptive optics. Because as you can see here, by comparing the AO off and on images, even if we denoise the AO off, we get a better contrast, but we cannot resolve the fine features of the glioblastoma cells. And this is, of course, also recapitulated once you do line plots across these membrane-labeled glioblastoma cells, where only with adaptive optics and then, of course, with denoising, we can nicely resolve them. So with all this in place, we were now ready to start some deep tumor imaging. And so Amre in my lab teamed up with Stella and Mark in Varun's lab, and they started to do deep brain imaging. And here's just a fly-through of a highly infiltrated mouse brain. And especially here on the bottom with the blue, you see the corpus callosum layers, where we wanted to do actually now time-lapse imaging to look at the cell migration patterns. So we basically park ourselves at around 1 millimeter here in this blue region in the corpus callosum. And we were able to actually observe many different invasion migration phenotypes. So most of the cells perform so-called microtube branching dynamics, but we also observe so-called translocation events and locomotion events. And this was important for our collaborators to actually compare how this is different from the cortex. Now, next we also wanted to better understand the invasion routes that these glioma cells could be taking, and here the white matter tracts and importantly blood vessels constitute a very important tumor microenvironmental niche. However, we also have to realize that both blood vessels and myelin, or the corpus callosum, give rise to the same third harmonic label-free signal. So we first had to come up with a way to unambiguously differentiate the two signals. And so to this end, we trained a customized machine learning algorithm based on elastic to actually distinguish between the myelin and the blood vessels, so that we could actually distinguish them. And so now this then allows us to actually see how the glioma cells actually fit into the corpus callosum fiber tracts, and how they actually use the blood vessels as an important invasion route, which you can see here on the plot on the left. And actually what our collaborators could tease out of these images is that there's a so-called orthogonal vessel migration route that these glioma cells are taking by branching out, grabbing some of these microvessels and then translocating the body. And also these migration patterns actually resemble a lot the oligodendrocytic and astrocytic precursor cells during development. All right, so with this I'm almost at the end of my presentation. I showed you today how three-photon microscopy allows us to increase the imaging depth, and how adaptive optics allows us to maintain almost diffraction-limited resolution. I also showed you some applications to functional and structural imaging inside the brain, both astrocyte calcium dynamics, but also longitudinal glioma cell migration imaging. And I've also wanted to take this opportunity to also actually tell you that our lab develops microscopy techniques in a very broad fashion. I only talked about a tiny fraction of what the lab is doing at the moment. We also have many projects that deal with high-speed volumetric imaging, but also with biophysical imaging based on brilliant microscopy and photoacoustics. So in any case, if you are interested, please get in touch. It would be great to hear about what you're all trying to do here. And last but not least, I also wanted to mention that in the field of high-speed imaging, we more recently are working with two techniques, optical coherence tomography and the oblique plane microscope concept. But especially here, I wanted to now just bring your attention to our work in optical coherence tomography, because we're actually having a demo here and a workshop. And so we are now developing optical coherence microscopy techniques that allow us to really look at organismal scale dynamics in a very fast manner. Here you can see applications to a lot of marine biology, Nematostella, Platinarays. Here you can see some sponges. And what we realized is that to really study marine biology in the field, we need to be able to miniaturize our microscope and actually bring it on site. And this is what we did. So Ling, a fantastic scientist in my lab, actually miniaturized this optical coherence microscope. He put it into a single traveling suitcase, and we brought it here on the train. It will later this year and next year be on the Amber truck that will actually traverse Europe's coastlines to study the marine and coastal biology across the scales. And I really want to highlight that we are having a demo here. We have a workshop number 147. We already did it last evening. We have another one this evening. So please, if you're interested, come by room 9 in the kids club. Or if you just have an interest in trying it out, please come and approach us. All right, so with this, I'm really at the end of my talk. I want to thank the fantastic people in my team, especially Lina, Ling, Emmanuel, and Amre, whose work I've presented here today. And in case you're looking for positions, we have a variety of positions open in our lab.
Thanks a lot for the impressive talk and material. My question concerns essentially the constancy, or otherwise, to put it differently, the variability of the adaptive optics correction. I mean, how much does it change from one depth to the other? First question. How variable is it in time? I mean, how often do you have to refresh it when you do imaging? And the third question is, how variable is it from one individual to the other? For example, could one say, okay, I have sort of an order zero order adaptive optic correction and apply this for all mice at a given depth? And
yeah.
Yes.
Yes, so I can comment on all of this. So first of all, I want to highlight that when I speak about adaptive optics improvement, this is really the improvement due to the tissue aberrations. So we do what we do in the field we call systems aberrations. We subtract them, so we basically don't show them. So basically, when you go from one animal to the next, you really have to reperform the adaptive optics. as I showed with some of my isoplanetic patch analysis, the aberrations don't vary very quickly in space. So this is on the order of about a few hundred micrometers. So if you think of a volume, you have to perform a correction measurement every few hundred micrometers. Now, how does this change in time? Well, we actually found that over the duration of a single experiment, which can last up to an hour or maybe more, there wasn't a big change. So it's really sort of more static aberrations that we measure or that we are able to correct. And I mean, finally, I want to highlight that here we're doing adaptive optics. So this is really like low order aberrations. This is very different from, let's say, wavefront shaping, where you're trying to correct higher order aberrations, which can really change on the second or even millisecond scale. So our type of aberrations are quite static in time. So we really only do it once and then perform the experiment. Well, the problem is still that we are having a scanning microscope, right? And so the frame rates are in the order of just a few hertz. So in that sense, voltage imaging would be very challenging.
Thank you very much for the talk. You mentioned that the noise of three-photon effects have to be taken into account a bit differently from usual. Can you explain a bit more what you exactly did in this denoising?
Yes, I don't think I brought an image how it typically looks like. Maybe one can see it here a little bit. There's a very structured noise that we often find, which is due to the fact that the PMTs have a so-called ripple noise, which we see a lot because we operate our microscope really sort of at the limit of the photon budget. And so this is one of the primary concerns that we see. And also then when we compared the noise distribution and statistics to other papers that have used noise to void for two-photon microscopy, we just found that our distribution looks a lot different. So that's the short answer. So you really have to sort of train your algorithm or your network to three-photon data. You can't just take one of the pre-trained two-photon networks and apply them.